Hello everyone, myself Deepa Singh, working as assistant professor in Gyan Vihar School of Pharmacy, SGVU Jaipur. Today we discuss about dissolution apparatus. Previous lecture we already covered different dissolution model. There's a tank dissolution model. According to this model, you check which model is suitable for your formulation or fit for your formulation. In some model, we can see that Higuchi model, Hexenproval model, diffusion layer model, first order, zero order model. So these are the different 10 models that we already discussed in previous lecture. These are the dissolution rate model. And with the help of this model or with the help of this equation, you calculate how much drug dissolve in particular equation or in particular formulation. And today we discuss about dissolution apparatus. These dissolution apparatus are very important to performing the dissolution test, because without these apparatus, you don't perform your test. And these apparatus are different types are available according to pharmacopoeia. According to USP, there are 7 types of a dissolution apparatus, and according to IP, there are 3 types, and BP is also 2 to 3 types of apparatus are available. In this lecture, we see different apparatus according to pharmacopoeia and their condition and how we perform these dissolution tests, the requirement condition, buffer, and rotation speed, and equipments, how that particular equipment work. So in this lecture, we covered all this parameter regarding this dissolution study of dissolution apparatus. This is the topic of biopharmaceutics and pharmacokinetics, 6th semester subject. And before starting this topic, you should know about basic object or learning object. In this learning object is this introduction of a dissolution apparatus, means the definition or introduction of part of the dissolution apparatus. What is a dissolution apparatus and how we perform this dissolution study with the help of this dissolution apparatus, and complete information regarding this dissolution apparatus. Next is type of dissolution apparatus according to pharmacopoeia. There's a different dissolution apparatus available according to pharmacopoeia. According to this pharmacopoeia, you know about how many types of apparatus are available for according to that particular pharmacopoeia, according to USP, IP, and BP, and their name and their procedure, how you use that particular apparatus. So types of dissolution apparatus according to pharmacopoeia. Condition and media for dissolution, which condition or which media is required to perform this dissolution state, and how we prepare this media for dissolution testing. Next is method of dissolution, how we perform this dissolution, or method and procedure of this dissolution study. That is all you study in this lecture or in this dissolution apparatus PPT. So before starting this topic, these are the apparatus that are according to different pharmacopoeia and according to USP, IP, and PP. In USP, there's a 7 apparatus. First one is basket type apparatus, or type 1 is a basket type. Type 2 is pedal type, and type 3 is reciprocating cylinder. Fourth, according to USP, type 4 dissolution apparatus is flow through cell. And according to USP, 5 type of dissolution is pedal over disc, and 6th is rotating cylinder, and 7th one is reciprocating disc. These are the 7 dissolution apparatus, or type 1 to 7, that is given in USP or a United States Pharmacopoeia for dissolution test. And these tests have a different construction or different working condition, and their rotation is also maybe vary, or particular apparatus you use for particular formulation, means every apparatus should have a different formulation for dissolution test. And these apparatus we discuss one by one in next slide. Next is IP dissolution apparatus. In IP, there's only 2 types of dissolution, that is pedal and basket. This is also in USP, but it's just opposite. In USP, first type is basket, but in IP, it's the second type basket, and pedal is second type in USP, but in IP, it's the first type, and just opposite. In USP, first is basket and second is pedal, but in IP, first is pedal and second is basket type. And in this BP dissolution apparatus, A is a basket type apparatus, means same the USP dissolution, or second is pedal. Second one is also similar to your USP classification, and third is flow through cell. In BP, there's a 3 types of apparatus only. Basket and pedal both are according to USP sequence, and third is flow through cell. So means in USP, there's a 7 types of the dissolution apparatus. In IP, it's a 2 type, and in BP, it's a 3 type. So this is all are the classification and type of a dissolution apparatus according to pharmacopoeia. You can select a particular apparatus for your formulation. This is selection depend on your type of formulation, which formulation you use for this dissolution study. So according to your formulation, you can select any one apparatus which is appropriate for your formulation. Now is types of dissolution apparatus according to USP. These are, now we discuss this one by one. Basket type, second paddle, then reciprocating cylinder, third one, then flow through cell, then fifth one is paddle over disc, and sixth one is rotating cylinder, and seventh one is reciprocating disc. Now this seven types of apparatus we discuss one by one in next slide, and we covered only these USP dissolution apparatus because in IP and BP all are seen, that is given in USP. Basket and paddle both are seen in IP as well as BP. The sequencing change in IP, first is paddle and second one is basket. But in BP, it's both are seen, and in BP there's a three, next third one is flow through cell, and in IP there's only two type of dissolution apparatus. Means all are similar, that is given in USP. So we discuss only this dissolution apparatus according to USP one by one. And in IP apparatus, paddle and basket, in this flow through the cell is one more, which is also same to USP. And first one is basket, and second one is paddle in BP. Now, initial feature of dissolution apparatus is a dissolution apparatus or proper construction of the dissolution apparatus. It will good result when you perform dissolution study of your formulation. And it's a buffer solution that you put in dissolution solution. When we see the construction of this, then you can say that this is a beaker type container. And in this, you put a buffer solution or your solvent that is required for dissolution testing of your formulation. And you put here tablet or your drug that is study for your dissolution test. And this is the chart after adding tablet, homogenization or mixing gets started. And you set a particular speed for this dissolution, and temperature is majorly required, body temperature for all type of the dissolution apparatus, because in this dissolution test you just considering your body for this dissolution apparatus. That's why we select 37 degrees Celsius temperature for this dissolution test, or almost all type of dissolution require 37 degrees Celsius temperature for testing. And after setting this temperature, the rotation gets started, and the tablet get disintegrate or any type of formulation will break, and slowly it distributed in this solution or in buffer solution. And after particular time interval, according to type of formulation, maybe 10 minutes or 30 minutes, you withdraw the sample from here. And suppose you are withdrawing 1 ml or 10 ml or how much, when you add fresh buffer to this solution, this is known as Singh condition. Means when you add a fresh buffer to this solution, that how much you remove, similar amount of you added in this solution, then the maintenance of this condition is known as Singh condition. So you should maintain Singh condition in this apparatus or in this solution. So this is all are the dissolution apparatus. Now we perform the test or perform the study. Now we start this. In this, fabrication dimension and positioning of all components must be precisely specified and reproducible. Means the dimension and component of these dissolution apparatus are properly set or precisely set or specified and reproducible in manner. And you can use this without having any problem or without setting any dimension, because it's a proper fabrication dimension as well as precisely specified. You can set the temperature and speed of this parameter in monitor and start performing your dissolution test. Next is must be simply designed, easy to operate, unsealable under a variety of conditions. And this dissolution apparatus condition or construction is also very easy. Anybody can handle when he or she knows that a procedure how you perform this dissolution test. The specified training is also not required. So the design is very simple. So you can easily operate this dissolution test and unsealable under a variety of a condition. It easily suitable or comfortable in variety of a condition or variety of equipment or pH condition. And next is must be sensitive to develop process change and formulation difference. Once you can say that it's a the good result, as well as when you change the condition, then it can easily evaluate result or process change of formulation different, but still it repetitive result and an identical condition. Means when you start performing this apparatus or handling this apparatus, it should be proper result or identical result and proper condition or repeated condition. Don't show any type of the error. Next is the apparatus in most cases should permit control but variable intensity. Means the drug is released in a controlled manner or variable intensity of mild uniform, not turbulent liquid agitation. Next is uniform flow is very essential because change in hydrodynamic flow will modify the solution. Your formulation should be uniformly run or uniform flow is very necessary so that hydrodynamic flow will be modified the resolution. Next is nearly perfect sink condition should be maintained. Means you should maintain proper condition or sink condition. Sink condition, we already discussed that what is the sink condition. Sink condition means how much buffer solution you remove from particular solution or particular buffer, and you add fresh buffer solution in your sample. So this condition also known as sink condition. You should maintain the sink condition for appropriate result. When you maintain the sink condition, it gives good result and less error or variation is observed in your formulation. So this is regarding the apparatus, and in this next, apparatus should provide easily a means of reducing the dosage form into the dissolution medium and holding it once immersed in a regular and reliable fashion. Means there's no specific condition or any technique is required for introducing a tablet or formulation into this dissolution. It gives good condition result or inverse result when you put your tablet or the dosage form introduced in your medium. So it easily handle or after immersion, it give proper or regular fashion results in your formulation. Next in this, you can say that it should be provide in this next is it should provide minimum aberration to the dosage form during test period to avoid disruption of a microenvironment surrounding the dissolving form. Means there's a less error minimum aberration is observed in the dissolution test during the performing this dissolution due to presence of a microenvironmental condition or microenvironmental surrounding of your formulation or your buffer solution containing that condition. So less number of aberration or error are observed or minimum disruption is observed in this formulation. And next is a special care should be taken that medium must not evaporate from container of the apparatus. Means if your formulation or your solution is in a volatile in nature or easily evaporate in nature, in this you should take some condition, a special condition is required that particular medium that it is not evaporate in a container or through that apparatus or dissolution apparatus. We should be in a proper stable form and don't reduce their volume or the volume is not less during the performing or during the taking this test. So you take care of this that your solution or your buffer solution or any other solution that you use for the solution should be in a specified volume and don't get evaporate. And next is samples should be easily withdrawn for automatic or manual analysis. Means your sample you can easily withdraw with the help of some manual or analytical procedure or easily take the sample without interrupting the flow characteristics of the liquid. Means flow characteristics is not affect of your formulation or your liquid. It easily withdraw with the help of some manual or automatic analysis technique. Next is apparatus should capable of allowing the evaluation of a disintegrating fluid tablet or capsule, finally powdered drug and novel solid dosage form. Means this apparatus should be capable or should have a proper evaluation done or allow to disintegrate your tablet or capsule. Means breaking of your formulation or tablet or capsule should be takes place, and after disintegration it can easily dissolve in solution and easily float on that particular solution. And finally powder drug and novel solid dosage form is used for this type of a dissolution testing. So your formulation should have a proper disintegration rate or easily disintegrate in a particular solution. In this, next is official dissolution apparatus. So these are the apparatus. There are 7 types of the apparatus. According to type, There's a drug formulation that is required for testing. In type 1 apparatus, first is rotating basket. We already see this type. And official apparatus in type apparatus 1 rotating basket. It is a conventional tablet, chewable tablet, and controlled release formulation. Means these are the formulation or these are the drug that are used for this apparatus 1 type or rotating basket type. This mainly use tablet or chewable tablet or controlled release formulation in rotating basket or type 1 apparatus. In second, apparatus 2, that is a rotating paddle. So in this second type, orally disintegrating tablet or chewable tablet or controlled release product or suspension use, there is almost all type of formulation are similar. There's little bit changes in this chewable and controlled release formulation are same for this, but it has orally disintegrating as well as controlled release suspension. Suspension is also used for this type of apparatus or paddle rotating. Next, third type of apparatus, that is reproducing cylinder. In the reproducing cylinder, use chewable tablet, controlled release formulation. This is also similar. In this, you use some chewable tablet or controlled release formulation. So third type apparatus, reciprocating cylinder. And next is type 4. This flow through the cell. Flow through the cell is majorly used for limited type of formulation, mainly poor soluble drug or poorly soluble drug, and powder granules, microparticles, means a small size particle or those drug who are poorly soluble, used or perform dissolution test by this type of apparatus, that is flow through the cell. Those drugs who don't have a proper solubility or poor solubility. And granules particle, that is also small in size, and microparticle, small size particle, you perform the solution testing by this apparatus 4. Next is apparatus 5. In this paddle over a disc, this is mainly only for transdermal formulation, or example in this is like transdermal patch. So those formulation who are transdermal available, you perform the solution with the help of this paddle over a disc. Next, type C, that is cylinder. And this also same transdermal formulation is required and perform the solution testing of transdermal formulation. Next element is reciprocating disc. In this reciprocating disc, controlled release formulation, transdermal formulation is performed. Means transdermal is mainly for last three type of apparatus, 5, 6, and 7. For transdermal formulation, you use this three type of apparatus, or out of this three, you can use any one type, which is appropriate according to requirement. And in last one, this controlled release formulation is also added. Hence, both transdermal as well as controlled release formulation are studied with this 7 type apparatus or reciprocating disc apparatus. So these are the example according to different apparatus or their name. Now we start the apparatus one by one according to their types. In this first type in USP, that is basket type, or USP apparatus 1 in basket type. In this vessel made from borosilicate glass, same spherical bottom capacity, that is 1000 ml. Means manufacturing or maybe formation of this type in basket type container with the help of borosilicate glass, or we use borosilicate glass for the vessel manufacturing of type 1, and same spherical bottom is there for this vessel. Means the bottom is semi spherical or half spherical, and capacity is 1000 ml for this type 1 basket apparatus. Next is shaft. Shaft is a stainless steel. 316 rotates smoothly without significant wobble. Means there's a one shaft. We already see the structure or diagram of this dissolution test. There's a one beaker like this is known as vessel, and the capacity is 1000 ml, and this made of borosilicate glass. Next is there the shaft that is dipped in that beaker or like structure of that vessel, and this shaft is mainly made of stainless steel 316, and this helps to move the solution or smooth rotation of your sample. It's a diagram of this type 1 or basket apparatus. In this basket, stainless steel 316 gold. It's your basket. And the water bath maintained at 37 degrees Celsius plus minus 1. We already discussed this condition. Means for this there's required 37 degrees Celsius of this temperature, because this temperature is your body temperature, and in dissolution test you use your body temperature or set according to body condition, because you are comparing this in between going to test. So temperature requirement for this water is 37 degrees Celsius, means this solution should temperature of 37 degrees Celsius plus minus 1. And used for capsule tablet from this suppositories floating dosage form. These are the example of formulation for this basket type. Means you can also use tablet, capsule, or maybe sustained release or controlled release, or suppositories or floating dosage form. These are the examples used for this first rotating basket type apparatus or USB-1 apparatus. In this, when we see the diagram, this is a vessel or beaker-like structure is known as vessel. It is made of a borosilicate glass, and it's a shaft. In this, you put your tablet, and it's a dosage form. You can see that it's a dosage form that is put in this shaft. After this, you start rotating at particular rotation, and temperature of this medium it should be 37 degrees Celsius. And after rotating, you withdraw the sample by here with the help of the pit, and how much sample you remove, you add fresh water in this. So you maintain sink condition, and this shaft is made out of stainless steel. So this is regarding the rotating basket. In this rotating basket, apparatus consists of cylindrical basket held a motor shaft. Means a cylindrical basket, you already see the figure, and there's a one motor shaft that rotate in the cylindrical basket. And second basket hold the sample rotation and round flask containing dissolution medium. Means it contain one whole sample or basket that hold your sample and rotate in the round bottom flask or containing dissolution medium. Dissolution medium is mostly a buffer solution. Next is entire flask is immersed in a constant temperature bath set at 37 degrees Celsius. Means that shaft or flask you put in the buffer solution at constant temperature, and that is 37 degrees Celsius, and start rotating constantly at 37 degrees Celsius temperature. And after the rotating speed and the position of the basket must meet the specified required set for in the current USP. Means rotating speed or position of a basket should be specified according to requirement, and you set this according to USP or according to USP requirement. This speed should be in a, specified in USP and should be in a constant condition, and most common rotating speed for basket is approximately 100 RPM. Means according to USP for type 1 or rotating basket type apparatus, you set the speed is 100 RPM, and start rotating this should be constant for overall procedure, or the solution is also constant for specified requirement. This is all about the type 1 apparatus. Next is type 2 apparatus, that is paddle type. According to USP, first is basket, second is paddle type. In this paddle type, vessel is seen as your type 1 apparatus, means made up of a borosilicate glass, and in shaft the blade passes through shaft so that the bottom of the blade fused with the bottom of the shaft. Means blades are or shaft are also seen, and these shafts are in a blade-like structure or looks like a blade. And due to this blade, it passed through the shaft and bottom of the blade fused with the bottom shaft. And next is a stirring element made of Teflon for laboratory purpose, stainless steel. There's a stirring element that is connected with shaft on attached with shaft. And this stirring element is made up of stainless steel or made up of Teflon for laboratory use. And next is water bath. Water bath is also similar to type 1 apparatus, in which you maintain the temperature is 37 degrees Celsius plus minus 0.5 degrees Celsius, because it's also, you can say that body temperature. So you maintain 37 degrees Celsius. Next is sinker. Sinker is mainly made of platinum bio, used to prevent capsule tablet from the floating. Means with the help of the sinker, a tablet won't float, or it can be stable at a particular desired site. So it helps to prevent the floating condition, floating of your tablet. For this purpose, use platinum wire, or sinker is made of platinum wire. This is a diagram of second paddle apparatus. In this, you can see that this is a dissolution vessel or rotating paddle shaft is this one, and it's a paddle. Name itself says that one paddle in this. This paddle is attached with this rotating shaft. You set this shaft to a paddle, and it forms one rotating paddle shaft. And this is a dissolution vessel complete. And in this After setting, you put your buffer solution or your solution that is required for your dissolution. And here's the setup. In this, it's a dye and gasket and plastic cap. And the drug content put in this gasket. It's your drug content compact or drug formulation. After putting this shaft or paddle get rotate and slowly your drug gets released. And with the help of this basket, your drug get released slowly, or it comes in the contact with solution. And after this, you remove the sample or the buffer solution, how much drug is present in particular amount, and maintain the sink condition. And with the help of you calculate how much drug released in particular time period. So this is a construction of a type 2 or paddle apparatus according to USP. This is another solution form of particle science of this apparatus 2 diagram. Next is the paddle apparatus consists of a special coated paddle that are minimized turbulence due to stirring. This paddle or this blade-like structure paddle has to remove the turbulence, and uniform flow is takes place with the help of this paddle. And a coated paddle that minimizes the turbulence due to stirring, and it consists of a special coating. And the paddle is attached variety of a variable speed motor that rotate at a control 40 speed. Means the speed of this type 2 or paddle apparatus is fixed or should be specified. And the speed is mainly in a control amount, but this 40 speed you set. In previous one, we type 1 we set 100 speed, but in this we set 40. And the tablet or capsule is placed in the round bottom of the solution flask, which minimizes the turbulence of the dissolution media. We see the previous slide in this diagram. You can see the sample that is put in this dissolution flask, and after we get start rotating, and this has to prevent the turbulence and dissolution of the medium uniformly rotate or uniform distribution. The apparatus is housed in constant temperature, water hall maintained at 37 degrees Celsius. And similar to rotating basket method, means temperature is similar for both E and type 1 and type 2 type, that is basket and paddle type. We maintain 37 degrees Celsius temperature for this. Next is the position and elegant of the paddle are specified in the USP. The paddle method is very sensitive to tilting. Improper elegant may destructively affect the dissolution result for some drug formulation. Means in this you take precaution during setting of this paddle or this shaft, because it set properly on sensitive way in an elegant position. If you don't set this in proper way or it should improperly set, then it shows some problem or should drastic effect or some error in your dissolution result of this formulation. So you set this shaft in a proper or specified elegant way. And next is most common operating speed for apparatus 2 is 50 RPM for solid oral dosage form and 25 for suspension. Hence, the speed is depend on type of the formulation. Type 1, that is in basket, we set 100 RPM, and type 2 we set 50 RPM, but this is also specified formulation. 50 RPM is only for solid oral dosage form like tablet and capsule. And if you are using suspension like formulation, then you set speed is 25 RPM is sufficient. Next is apparatus 2 is generally preferred for basket, and a sinker such as a few turns of platinum wire may used to prevent capsule or tablet from floating. Majorly this type 2 apparatus is used for dissolution because it gives good result, and there's a one platinum wire is present in the dissolution. So this platinum wire has to a stable form of the tablet and protect the tablet or capsule to floating, and it is prevent this turbulence in floating. So this is mostly preferred for tablet and capsule type of dissolution, and shrink condition is also maintained in very easy way. Next is type 3 recipe apparatus, that is reciprocating cylinder. This type 3 or reciprocating cylinder, in this design, vessel is cylindrical flat bottom glass vessel. Means it's a cylindrical shape and bottom is flat or flat glass vessel bottom. And agitation type is reciprocating, generally 5 to 35 RPM. Means rotation is normally 5 to 35 depending on the type of the formulation. And volume of the dissolution fluid is 200 to 250 ml. There's a volume is very less that is required for a dissolution. In type 1 and type 2, you require at least 1000 ml or 1 liter volume. But in type 3, you require only 200 to 250 ml volume. for this dissolution test. Next is water bath. In this, maintain 37 degrees Celsius temperature or plus minus 0.5 degrees Celsius. This same temperature is required. And use of this is mainly for external release type of formulation or use for external release formulation. Almost some parameter are same. In this temperature is same, but volume is 2 to 250 ml. In previous one there is 1000 ml, and RPM is 5 to 35 is sufficient for type 3 apparatus. In previous we used first one is 100 RPM. Next in 50 for tablet, for suspension is 25. In third apparatus we use 5 to 35 according to different type of formulation. It's a diagram of apparatus 3. It looks like cylinder or reciprocating cylinder and flat bottom. In type 1 and type 2, it is a semi-spherical bottom, but in type 3, it's a flat bottom. And here a dosage form, or this cylinder contain dosage form. And after this, you start rotating, it release slowly in your solution. You can see this. You have to start releasing, and after releasing, you take the sample and maintain the same condition. And with the help of you calculate the percent drug release. And the USP apparatus is considered the first line apparatus is product development of control release preparation. Means when you are preparing control release formulation or taking the readings of dissolution. Use dissolution apparatus is your type 3 apparatus. Majorly use type 3 apparatus for control release formulation. And in this, in product development of a control release preparation, because of its usefulness and convenience in exposing product to mechanical as well as variety of physical chemical condition, which may be influence the release of product in the GI tract. But it show good result and conveniently to use. Some physical chemical condition may be affect this dissolution rate and affect the product rate in GI tract. And particular advantages of these apparatus is technically easy and problem-free use test solution with different pH value for each time interval. Means advantages of these apparatus is also good, and for control release you can easily use it won't show more error or problem-free test solution is obtained when you use this type of a dissolution. And pH of value is also maintained particular time interval. And it is also avoid concentration formation of a disintegrate or immediate release type of a tablet product. And when we encounter DSP apparatus too, so this is the more suitable for immediate type or control release type formulation when we compare type 2 or type 3 type apparatus. And this is regarding the type 3. In this additional advantages of type 3 include feasibility of a drug release testing of a chewable tablet. And chewable tablet for human use do not contain disintegrant, so that need to undergo physical grinding, that is chewing prior to dissolution. Means you can also use this type 3 apparatus for chewable tablet. With the help of this type 3, you get good or proper result of chewable tablet and disintegrate in physiological grinding or chewable before prior to dissolution is example. And this entry coated or sustained release forms or also affect the advantages of mimicking the change in physical chemical condition, extraordinary strong medical forces experienced by drug product in the mouth or at certain location in GI tract, such as pyrolus and equilateral wall. Means this type is mainly used for both entry-coated and sustained release type of formulation, because the tablet get released in the intestine or in GI tract, that is pyrolus and equilateral wall, and it show advantages of mimicking the changes of physiological condition and show good mechanical forces to the product form in particular condition. So this is the advantage of one example of this type 3 apparatus. Next is type 4 apparatus. In type 4, that is flow through the cell, and in this design, reservoir for dissolution medium. Pump is forced dissolution medium through the cell holding sample flow, and 10 to 100 ml per minute laminar flow is maintained. Means there's rotation is takes place, so continuous rotation takes place in this apparatus with the help of laminar flow or laminar. And speed is 10 to 100 ml per minute for this medium flow. So the cell holding, and reservoir is for dissolution medium, and water bath is maintained 37 degrees Celsius temperature plus minus 0.5 degrees Celsius. Means in this condition you also maintain 37 degrees Celsius. This temperature, this 37 degrees Celsius temperature is common for all type of formulation or all apparatus of USP, including type 1 to type 7, because this is a body temperature. It's a diagram of four type of flow through the cell. And this one is the cell body or upper part of your apparatus, and it's an outlet for flow, and it's a gasket. Means after cell body, there's another part is known as gasket, and this is RF coil and a strainer, and HBS is this one. And next is glass beads is this. This is a glass bead or bead that made up of glass, and this is a ceramic base, and next is cell body or lower part. So this is the upper part, this is the lower part, and this between this glass beads are present, or RF coil or stainless HBS is also present in this, and it's the outlet and it's an inlet. You put your sample from here, and your sample is here, and this is the outlet for the flow. Next is it is a two type. One is open system, second is closed system. In open system, the open system has a configuration where fresh medium is pumped through the cell and friction are collected. Means you continuously add a fresh medium with the help of this open system and pump out this medium with the help of cell or with the help of some friction. And next is closed system. Closed system is to maintain the same condition, but in open system you continuously replace the solution, or it is easy to add the fresh medium in open system, but in closed it should be in configured of closed system, or you do not replace this medium in a particular equipment or in that equipment. So it is difficult to replace the fresh medium. It is closed in type. So these are the two types for this apparatus. Next is apparatus 5, that is paddle over a disc. In this vessel, shaft and steering element are similar for your previous one. It is made up of stainless steel for shaft, and vessel is for borosilicate glass, and steering element is also there for this paddle over disc. And in this sample holder disc assembly that hold the product in such a way that release the surface in parallel with paddle. Means your assembly side like that your drug is released parallelly or onto the surface of parallel way in paddle, and paddle is directly attached over disc assembly, and paddle is directly attached with your disc or with your assembly or your apparatus, and sample are dropped between the surface medium and the top of the paddle plate. Means in this type 5 apparatus, the placing of a sample is also easy. You can easily replace the sample with the help of top of your paddle over disc or with the help of this apparatus. And volume is 900 ml for this disc or disc apparatus, and temperature is 32 degrees Celsius. In this temperature is less required, that is 32 degrees Celsius. Next is apparatus 6, that is rotating cylinder, or USP apparatus 6 is also known as rotating cylinder, or name is rotating cylinder. In this vessel, in the place of a basket, cylinder is used. Means in previous one we used basket, but in this basket, cylinder used in the place of basket. And cylinder is mainly made up of stainless steel 316. This is also made up of stainless steel. And sample is mounted to chlorophane, that is inner porous cellulosic material is being made. And next is entire system is adhered to the cylinder. And dosage unit is placed in cylinder and released from the outside. You use your dosage form or put a dosage form in the cylinder, and it releases slowly from the cylinder or outside of the cylinder. And release is very simple. And in fourth is the water bath. In the water bath, maintain at least 32 degrees Celsius temperature. Plus minus 0.5 degrees Celsius. Means type 5 and type 6 require 32 degrees Celsius temperature. Other remaining is required 37. But in 5 and 6, you maintain 32 degrees Celsius temperature for water bath. And next is disadvantages. In this, transdermal patches can be studied but cannot be cut into small size. Means you use transdermal patches for this type 6 or type 6 apparatus for USB, and in this use this transdermal patches, but you cannot cut this transdermal patches in a small size. Use entire patch for this dissolution study. This is the one advantage. Disadvantage of this rotating cylinder type 6 apparatus. It's a diagram. In this, it's your rotating cylinder, and the dosage form release from this side, or it's your patch that contain the drug, and it's your water bath of buffer solution containing, and you put your patch here, and it release slowly through this beaker or through this apparatus So this is a diagram of type 6 rotating cylinder. Next, seventh or last apparatus is reciprocating disc or type 7 apparatus. It is known as reciprocating disc. In this vessel is flat bottom cylindrical vessel, means bottom is flat or cylindrical vessel in shape, and volume of distribution is mainly 50 to 200 ml. Now to maintain the volume is 50 to up to 200 ml volume is required for this vessel. And shaft is mainly made up of a stainless steel. And next is sample placed on the disc shape on the holder. Once you put your sample on that holder or that reciprocating disc shape holder. And agitation is reciprocating or the reciprocation frequency is 30 cycles per minute. Means in 1 minute it covers 30 rotation or 30 cycles in 1 minute. So it's a rotation for this reciprocating disc, and maintained temperature is 32 degrees Celsius plus minus 5 degrees Celsius. Means you can see that out of the 7 apparatus from starting 1 to 7, starting to 1 to 7, some apparatus require 37 degrees Celsius temperature, some require 32 degrees Celsius. And we see that first to 4 or apparatus 1, 2, 3, 4 require 37 degrees Celsius according to body condition, but the apparatus 5, 6, and 7, last 3 apparatus require 32 degrees Celsius temperature, because in this mainly we use transdermal formulation or patches type of a formulation. That's why you use 32 degrees Celsius temperature for last 3, that is 5 to 6 type. 5, 6, and 7 type of apparatus, but first to 4 use 37 degrees Celsius temperature, and uses as for same is the transdermal patches for this reciprocating disc. It's a diagram of this reciprocating disc. It's like a beaker or cylindrical in shape, and there's a flat bottom that is spherical corners, and in this one is a disc where you put your sample or your patch in this disc or onto this disc, and this is a dissolution media or buffer solution containing vessel. You put a buffer solution, and on the disc you put your maintain constant temperature of this dissolution medium or buffer medium is 22 degrees Celsius, and after that your drug is released slowly from this patch at particular time interval, and rotation is I think 30 rpm, or not 30 rpm but 30 cycle per minute is required for this type 7 apparatus. So this is all regarding the USP apparatus that is required for the solution, starting from type 1 to type 7 apparatus. These are the 7 apparatus for USP, or these apparatus are according to USP, and according to BP and IP both are same procedure, just sequence or name of this apparatus get changed. After completing this topic or dissolution apparatus, we should know about learning outcomes. In learning outcomes, introduction of the dissolution apparatus means what is the dissolution apparatus and how we use the purpose of this dissolution apparatus. Next is type of dissolution apparatus according to pharmacopoeia, means different classification of these apparatus according to USP, BP, and IP, and condition in media for the dissolution, because every formulation requires different condition or different media, maybe some temperature changes, pH or buffer cell changes. So you know complete information regarding this condition in media for dissolution. Next is method of dissolution, means how we perform this dissolution test or different method for this dissolution. So this is about the learning outcomes. After this learning outcome is the reference part. For this topic, use all reference or single reference is sufficient. In the reference, first is Alton Pharmaceutics, the design and manufacturing of medicine. Third edition, page number is 17 to 22. Next is one Wikipedia site, https /un wikipedia.org/wiki/dissolution testing. It's a one site for Wikipedia. In this International Journal of Current Biomedical and Pharmaceutical Research, Current Science Direct Publication. It's another paper for this test. And fourth is USP 30 and NF25, US Pharmacopoeia Convention 2007, Blackwell MD. So these are the good reference for this topic for dissolution apparatus. After this, we covered the question part, which is similar to this topic. In this first is dissolution test apparatus. Number 1 as per BP is 1 is paddle, is basket, C is rotating cylinder, and D is rotating cylinder. And C is basket and B is cylinder. Out of this, you can any one correct option according to BP or type of classification according to BSP, BP or British Pharmacopoeia. In second question, USP7 dissolution apparatus is extensively used for evaluation of a tablet. B is capsule, C is transdermal patch, and D is the all type of formulation. We already covered this part in this PPT. So we will answer out of this ABCD option. Third is a dissolution test apparatus number 1. As per IP is A is paddle, B is basket, C is rotating basket, D is rotating cylinder. So in this, you select type 1 apparatus according to IP. And fourth is which form of the drug show rapid dissolution rate. There is no about the formulation type. A is crystalline, B is amorphous form, C is hydrate form, and D is a solvent form. According to this, you select out ABCD option which have a more dissolution rate. Next is in fifth question. In heavy or weaker method, stirrer is rotated at the 69 RPM, 59 RPM, versus 79 RPM, and D is 49 RPM. So these are the four options for fifth question. And in sixth question, paddle over disc apparatus is extensively used for the evaluation of. First, you know about what is a paddle over disc and which pharmacopoeia contain this and the type. And after this, you know which parameter or which type of formulation used for evaluation by the help of this paddle over disc. In this, A option is tablet, B is capsule, C is transdermal delivery system, and D is all type of the formulation. So these are the type of formulation that is required to perform the dissolution test with the help of paddle over disc. Out of this, you can select any one correct answer. Next is the official dissolution test apparatus contains electrical vessel and lower the edge of a plate is positioned from the inside bottom vessel. In this 18 to 22 mm, B is 22 to 27 mm, C is 22 to 24 mm, and D is 25 to 29 mm. Basket and this rotating cylinder. In this, you select apparatus type number 1 as per USP. Out of this four option and even that option. Last is 10th question. In 10th question, the solution test apparatus number 7 as per USP is means you know about different 7 apparatus according to USP, the name, and according to USP, you can select any one correct option out of this ABCD option for USP 7 type of apparatus. What is another name of this apparatus? There is the option pattern. There is a basket, C is reciprocating basket, and D is a reciprocating disc. So these are the 10 questions related to this dissolution apparatus or this topic. So we covered already different type of dissolution apparatus according to different pharmacopoeia, including question. So thank you so much for watching and listening to this video. Thank you.